How do we get from a very simple 2 amino acid peptide to a complex protein like this? Well, now we need to start talking about protein folding and different levels of protein structure. So, protein structure has 4 different levels. And the first level of protein structure is just the identity of the amino acids. So we already have talked a little bit about this. The identity of the amino acids is important because different amino acids have different chemical characteristics. They can be polar, nonpolar, electrically charged. We've also seen that some amino acids have larger side chains than other ones. So glycine, again, it just has the hydrogen, very small. Other amino acids have quite large side chains. So the primary structure is crucial in order to know the function and to predict the folding and the function of a peptide. The second level of structure is the secondary structure, easy enough, and this refers to either alpha helices or beta pleated sheets. Oh, so what are these? Well, an alpha helix is what we see here, and an alpha helix can form between many different types of amino acids. It depends on hydrogen bonding, which results in the curling of a string of amino acids, of an amino acid polymer or a peptide. So this is a nice little diagram here. So it shows that there are intra-chain hydrogen bonds, which means that there's hydrogen bonding, not between the R groups, but between the amino groups and the carboxyl groups. So we see that occurring between many amino acids. And what did we learn about hydrogen bonds? We learned that they are weak, but oftentimes we see many of them in biological molecules, right? So each individual hydrogen bond, the dotted bonds we see here, is weak, but this whole protein is held into this alpha helical shape because there are lots of hydrogen bonds stabilizing the structure. And then I want you to notice here that the side chains can extend outward in this type of structure, which is, again, an alpha helix. And then we can look at it from the top, and we see that the side chains can stick out. And oops, this is a little bit off center. There we go. And so our side chains, our R groups, can stick out, and they might have different properties depending on which side of the protein they are on. So if we look over here on the right, we see the larger part of a protein, and then we see a small part of the protein highlighted at the top here. And so perhaps these blue R groups are actually hydrophobic, but the red ones are hydrophilic. And so this is a feasible structure for protein folding, because this means that an alpha helix has formed. On one side of the alpha helix, if you're looking down it as we are on the left, all those R groups would be hydrophobic. And then on the other side, the R groups would be hydrophilic. The hydrophobic side is associated with the rest of the protein, so it's not interacting with water. And then the hydrophilic side, as we see these red groups, are pointed out towards the exterior of the protein, so it's interacting with water molecules in the environment, potentially. All right, so the other type of structure in secondary structure is a beta-pleated sheet, and an example is shown here. So beta-pleated sheets are chains of amino acids that have 3D structure. They have these loop structures where we see, if we follow the linear chain, we can go up, we see that there's a loop, and then it turns back on itself. So again, we're seeing hydrogen bonding between the hydrogens and oxygens. So the hydrogens of amino groups, there we go, the hydrogen of this amino group, and the oxygen here of this carboxyl group. And we see it all throughout this beta-pleated sheet. So we see one here too, and here it's a little bit hard to see. And then this image also points out that the peptide bonds are what are holding the amino acids in a chain. So let me just make a note here that the linear structure, the primary structure of amino acids, deals with those peptide bonds. Hydrogen bonds are very important in the formation of the secondary structure, which are the two structures that I've pointed out here, up here and on the previous slide. And so what we see on the right here is that you can have beta-pleated sheets that are stacked one on top of another. So you'd have this whole structure that would be packed on top of another structure. And the R groups can stick out to the side and interact with other molecules, other parts of the protein, etc. All right, so that's secondary structure. Then our tertiary structure, our third level of structure, is the entire 3D shape of the protein. So in the secondary structure, we only have specific parts of the protein folded. In the tertiary structure, we have the entire protein fully folded. So tertiary structure depends on a number of different kinds of bonds. It's not just one type of bond like we saw for the first two levels. There are disulfide bonds between cysteine amino acids. There are hydrogen bonds between the R groups of these other amino acids. And we see ionic bonds, again, charge attraction between electrically charged side groups of amino acids. And then we also see hydrophobic interactions. And that is just the bunching together of any nonpolar amino acid side chains. So nonpolar molecules tend to clump together in an aqueous environment. The cell is an aqueous environment. So again, let me just specify that this purple thing, this purple line, is the backbone. So as we go through here, we'd be seeing the central carbon, we'd be seeing the carboxyl, we'd be seeing the amino group, and then repeating that over and over again for different amino acids. And then the things that are shown in the middle here and at the left, these are just the side chains of the amino acids. I want you to pay special attention to the disulfide bond. The disulfide bond is really important because it can form spontaneously. So that's really good for your proteins if they become unfolded. Why would they become unfolded? Lots of reasons. Chemicals, heat, sometimes even physical manipulation could cause your proteins to become unfolded. And when they're unfolded, then they don't have the same function because function and structure are closely linked, right? So we change the structure, then we mess up the function. You don't want your proteins to be unfolded, trust me. Okay, so disulfide bonds can reform spontaneously because these sulfurs are so electronegative and they're much more stable if they are bound together than if not. And then that can help to position the rest of the protein so that it could be refolded as well. All right. So finally, the fourth level of protein structure is the quaternary structure. So if you kind of compare this to the tertiary structure, you see that we're taking the tertiary structure that we see here, and we're just sticking it over here. And then we have 3 more copies of that exact same thing. Not all proteins have a quaternary structure. Not all proteins have a fourth level of structure. Some of them just have a 3D structure. They just have the tertiary structure and the secondary and the primary. They don't have that last level of structure. Proteins that have quaternary structure tend to be larger, and they're made up of multiple pieces, multiple proteins, but each protein by itself wouldn't be functional. And so when you bring all these guys together, that's when you have a fully formed functional protein. So this, for example, I think that this is hemoglobin, which has multiple subunits. And we see one of the subunits here in the tertiary structure. And then we have 3 other copies of that when we see it placed here with the 3 other subunits. Okay. So I'm just going to go quickly through this. This is an example of protein structure. using insulin, so here we see the primary structure, the order of amino acids. You could start to look at this and start to think which parts of the protein might be hydrophobic or hydrophilic, depending on the polar, nonpolar, electrically charged amino acids. And then we have the secondary structure. So these are some alpha helices, which are found in the protein. And just a reminder of what an alpha helix is here. Then we have the tertiary structure. So especially this protein has multiple disulfide bonds, which are shown in yellow. And these are very important for keeping the protein folded in its 3D structure correctly. And then you can have a quaternary structure, which involves multiple insulin molecules. A single insulin molecule might be slightly less functional than the quaternary structure. For the most efficient function, the highest function protein, you need to have the other pieces of this protein present. Something else I wanted to point out are the polar and nonpolar amino acids. So if you look at these two images, they might seem a little bit just abstract. On the left here, though, and the right, we have the same protein. So these two pictures are of the exact same protein in the exact same position. The only thing that's different is that the nonpolar side chains are highlighted on the left, whereas the polar side chains are highlighted on the right. What we would expect to see if we overlaid these two images is that the red side chains would be on the outside and the gray side chains would be on the inside. And so I'm going to try and draw that on the whiteboard. I don't have a gray marker, but I do have a black marker. So our nonpolar amino acids and their side chains we expect to be in the middle of this folded insulin molecule. I'm not going to get the real structure of it, but if you could see that here, I'm going to try and enlarge this again. Okay, there we go. So this is our insulin, only showing the nonpolar amino acids. And then our polar amino acids would be on the outside. So they would be encasing the nonpolar amino acids, essentially. So these are polar. And so they would be on the outside of the protein. The nonpolar ones would be on the inside of the protein. If you think about, I don't know, a tennis ball, or no, better yet, how about some deep fried ice cream? That's still a thing, right? So in deep fried ice cream, the outside is like the crust, and the inside is the actual ice cream. So the crust would be the polar amino acids, and then the ice cream in the middle would be the nonpolar ones. They don't like to interact. And very similarly, you could make the analogy there that the ice cream it doesn't like to interact with the environment because it's going to melt, and so the crust is kind of protecting it from the environment. It's the exact same way with a protein, because the crust or the polar amino acids on the outside are better at interacting with water than the nonpolar amino acids on the inside. So hopefully that analogy clears things up a little bit. All right, so moving on. Actually, I'm not going to talk too much about structural motifs. We will talk about these a little bit in a couple of problem sessions. So I'll try and focus on what we'll need for those problem sessions. So structural motifs are basically shapes that can be found in a lot of different proteins. And so I try and make analogies. It's not what the shape is made up of, so it's not the identity of the amino acids, but rather it's the shape itself. So there are lots of different types of wheels. We see wheels in lots of different machines. We see wheels on cars. They're made out of rubber, or actually I don't know what they're made out of nowadays, but we'll just say rubber. But then we also see wheels on carts. the bottom of my whiteboard has plastic wheels, but we also see wheels that are used for different purposes. So I have a ceiling fan here. It has some kind of motor that has something like gears, but which are essentially wheels. And again, they are made out of something different. But they are all these round structures that, if you really broke it down and looked at just the wheel part, they would be very similar shapes overall. So similarly, we have proteins that are similar in their shape, essentially, even though we see that they're made up of different amino acids. So this is one common structural motif. This is a zinc finger. So in this shape, the characteristic parts are that we see these little blebs that poke out of the protein, and they are associated with zinc ions. And so specifically in this case, we do see 4 amino acids that have to be consistently and they need to be included because they are actually interacting with the zinc ions. And so these are the cysteines. But the rest of these amino acids can vary in their identity. The zinc finger is really good at interacting with DNA because, as you can see, it kind of has a structure that can reach into DNA and wrap around it, as you can see here. Then we also have the helix-turn-helix, which is another structure that is used in proteins that interact with DNA. And again, we can see here that it could kind of curve around DNA and interact with different parts. And the characteristic parts of a helix-turn-helix motif, focusing on the green here on the left, we have some kind of helix. So again, think about the alpha helix that we talked about in the secondary structure. And then we have a turn. And so that is just a loop, a string of amino acids that connects this helix to another helix. And that's it. But again, the important part of this is that the positioning of these 2 helices allows them to interact with 2 areas of DNA at once. This area of DNA up here, and then perhaps this area of DNA or this one down here. All right. I also want to just quickly note that there are also random coils or unstructured regions in some proteins. Scientists used to think that these were useless, but actually we now know that they do have functions. I'm not going to talk too much about it because they're still not very well studied, but we know that they are encoded in the genome for a reason. Okay. Let's talk a little bit about protein folding. Yeah. So protein folding happens in steps. Scientists once theorized, some scientists theorized, that proteins would just fold randomly. So you would have this long string of proteins and it would just come into all these different conformations randomly until it settled in its correct state. We now know that's not true. If it were true, it would take forever for proteins to fold, and some of them probably would never fold at all. So instead, now we know that there is a gradual, stepwise folding of the protein that's important. Now, you don't need to memorize all of these steps, but all you should know is that the protein becomes more and more stable at each step, and that helps it get to the next step. So up at the top here, we would have a linear protein that is starting to fold into alpha helices and beta pleated sheets in the appropriate regions. And then we're starting to get 3D structure as we move down into this molten globule state. And then finally at the very bottom, that is where we see the final correct or functional protein structure. What I want to point out is that there are different final structures that are possible, and proteins can get stuck in the wrong structure. Why can they get stuck? Well, that is because these are stable conformations. So if we see here on the left, that is the energy, free energy. So when a protein is completely unfolded or almost unfolded. It's very high energy, which means it's unstable and likely to change shape. As we go down here, the protein is changing shape, but parts of it are becoming more and more stable. So the energy decreases. Finally, here at the bottom, the protein might find a stable shape that is not functional, not as it should be. And there are certain molecules that can help the protein get back into its correct shape. And so this is one of those proteins, heat shock protein 60. Heat shock proteins in general are a group of proteins which help to fold other proteins. I know it's a little bit confusing. It's like the chicken or the egg, but for this concept, just imagine that these heat shock proteins have been formed already, and then they're helping a new protein to fold. So we're seeing a new protein or a peptide chain being released here, or built rather. So heat shock protein 70 here is a small heat shock protein, and it binds to this peptide chain as it gets longer and longer as amino acids are added, and it just stops it from getting tangled. Pretty simple. Heat shock protein 60 is a chaperonin, and it has a completely different method of working and helping proteins fold. So HSP60 is like a little barrel, and so the unfolded protein actually goes inside of the barrel, which is HSP60, and there's energy that is needed, and there's actually another protein which is the top to the barrel, and then the protein gets inside there and it's isolated from anything in the environment which might be a bad influence. Like it's isolated from water molecules, it's isolated from other proteins that might cause it to misfold, it's isolated from charges. So this is the same idea of, you know, if your parents don't like your friends and you're like, I don't want you to hang around with them anymore, and so they isolate you so that you then fold up and have your proper function. I don't know. But while this unfolded protein is inside of the barrel, then it's allowed to achieve its normal functional folding pattern through this whole system that we just saw. This system is where it becomes more and more stable, and eventually it's released as a folded protein. And then the HSP60 barrel protein can be used again to refold another protein. So that's pretty neat. And just in closing here, I want to emphasize how difficult it is to determine the 3D structure of a protein just from its primary structure. If you only have the primary structure of a protein, that is the identity of the amino acids, it's very difficult to figure out the 3D structure. You might be able to identify certain areas as most likely having an alpha helix or a beta sheet structure, but figuring out the entire protein shape is really, really hard. So this is one of the reasons why we still have many genes in the human genome and in many genomes that we don't know what they do. We have the nucleic acid code. We can figure out what amino acids are coded for, but then what do those amino acids form as a protein is unknown. It's very hard to find the shape, and even if you find the shape of a protein, it can be difficult to find the function, so we won't even go into that. But that is our lecture on amino acids and proteins today, and I'll see everyone tomorrow for our lecture on DNA. Don't hesitate to reach out. I hope that everyone is doing well and enjoying the sun. Hopefully across all of our time zones we have some good weather. Bye.